today is about cell migration, application spotlight that we choose for today. So let's start maybe with some basic definition. What is cell migration and how cells actually can move? So cell can move due to cytoskeletal plasticity and its reorganization. There are different types how cell can do that. And one special type is moving with some flagella or cilias. That's typical for the prokaryotic organisms, but in the eukaryotic cells, it's also a case for the sperm cells. And why do cell move, and why is it important to study them? Cell movement, it's one of the key processes in their lives because it ensures that cell maintain the function of its tissue. For example, in epithelial cells, cell move because they want to heal something. That's the case of the immune cells. Cells also move because they want to divide, they want to grow, and especially in embryogenesis, the translocation of cells is really crucial for the proper development of the embryo, also angiogenesis. And some of the cells actually move, move, and we are not happy about it, and that's the case of the metastatic cancer cells when they want to invade to other locations. All these processes are really important for proper functioning of the tissues, and in case of cancer cells, this is the question of life and death. So it's a big part of the research in cell biology nowadays to study the cell movement. If you want to study the cell movement, precise cell tracking is the key here. And to precisely track some objects in the images, you need precise segmentation to precisely know where the object actually lies. And this is the strong application area for quantitative phase imaging data because of the high quality and high contrast and uniformity in time, especially. When you want to describe the cell movement, you can find in many papers really a lot of different parameters how to describe the cell movement. The traditional ones are, for example, describing the total path length or the Euclidean distance or, of course, the speed. Then there are some parameters describing meandricity. You can find in different terms for the same actually characteristics. And the data are usually expressed at the end as a motility rows diagrams because they express both the path length and the directionality. So a few examples that were actually monitored with our microscope. So this is the study of sperm cell motility done for the Reproductive Medicine Center. You can see a short video of the sperm cells moving, but some of them are not. And you know that this is the big question today, studies of reproduction diseases and or malfunctions. So if you want to study the cells, the sperms that are not moving, first you want to segment them, track them, and then gate them. And this is something that you can do even easily in our software when you simply plot the data for the speed and you select a gate, select the threshold. So only the cells that are not moving and their speed is below some threshold that you set are considered in that gate, and then you can correlate this to other morphological parameters and find the reason why they are not moving. Is it because they are more circular or because their mass is too high? So this is the analysis of the sperm population, for example, that was unable due to the precise moving and tracking, segmentation and tracking. Another cells that are quite motile are lymphocyte, immune cells. And here is another. Notice that it was the same magnification with our microscope, but the cells are significantly larger than sperm cells, even though they are still small compared to the other cells in our body. So this is lymphocyte imaged with the 20x magnification, the acquisition speed of 1 frame per second. You can see that each cell carries the unique ID number, and then the trajectories are drawn. So then we can study the data at a single cell level, so we can plot the path length for each of the cells. We can also plot easily in our software the motility diagrams, and then study, for example, less motile or more motile cells. Lymphocytes are important because they interact with the target cells, with the cancer cells. There's a lot of studies on this topic nowadays. So this is the video, actually the co-culture of the prostate cancer cells and the lymphocyte T cell attacking this cancer cell. In this time-lapse video, you can see that even though the lymphocyte is much smaller compared to the cancer cell, it's really effective in the protection, and at the end of the video, the cancer cell dies. No, it died. If you can precisely segment the cells and plot their trajectories, it's like spying without actually touching, without labeling, so you can spy actually on their interactions. So in this paper, there was also, in this research, it was also done with our microscope. Scientists were treating a cancer cell with some oxidative stress conditions, and they found out that some of them were actually resistant to this condition because they used engulfing. It's like cannibalism type of behavior when one cell engulfs the other one. And they were also studying what happened before the engulfing, how the cell was actively moving to the other one. And this enabled them to sustain much longer, even in really unfavorable conditions. So this could be the mechanism of some cancer cell resistance. Drugs that are directly linked to the studies of cell migration are called migrostatics, and the definition is that they are drugs interfering with all modes of cancer cell invasion and metastasis mechanisms. And again, to be able to precisely evaluate the effect of the migrostatics, you need a precise segmentation and its tracking. So another example of one of our users from Marketa, she's studying the microstatic effect of different cancer cell lines, and she uses, for example, motility row plots to evaluate which of the tested drug has the highest effect on the migration of the cells. And again, these motility parameters can be directly in our software correlated with other morphological parameters. And one special advantage with our microscope is that it uses incoherent light source, and that gives us the possibility to image cells and their movement also in the scattering media. Scattering media can be, for example, collagen scaffold or matrigel, something that is really diffusive and would be difficult to image with the laser-based quantitative phase imaging microscopes. The optical principle why we can do this with the incoherent light source is that the only ballistic light in our case is imaged. So all the diffuse light that was scattered to some different planes actually does not contribute to the hologram. It does not meet the reference arm and doesn't create a hologram. And this effect, this possibility was actually used in the research of one of our users when they were studying the different types of movement of cancer cells in collagen matrix. And they nicely described the amoeboid and mesenchymal type of the movement of these cells. So how is actually a cell moving the dry mass, the picograms of biomolecules from the rear to the front when it's moving through the collagen, if it's squeezing through the holes or if it's decomposing the collagen fibrils. And you can see on the images, the gray ones are actually the QPI data, but the colored coded ones display the difference between two following images. So the red color shows the parts of the cell where the dry mass increased, and the blue color shows the decrease of the dry mass. The areas where the dry mass was retracted from to the areas where it was pushed to the red ones. So even though you cannot tell it from the gray scale QPI images, if you use this subtraction and color coding, it nicely visualizes the differences and what is the mechanism of the movement of those cells. So let me summarize once again the main advantages of quantitative phase imaging technology, because that's the key technology of today's webinar. First of all, it's label-free technology. There is no label toxicity, non-invasive, non-disturbing monitoring of cells. It provides quantitative data about cell dry mass quantification and its distribution across time. It's of low phototoxicity that allows to perform live cell imaging in long-term experiments. So, for example, in the studies of the drug, lower doses can be applied, which could be more in vivo relevant. There is no photobleaching, so the data are of uniform quality over time, and therefore it can be more reliable. The acquisition of the microscope is really fast. It's a single-shot imaging technology, so from one hologram you get one QPI image. And the data are of high contrast, so with no background artifacts, no coherence noise, that allows to perform precise segmentation, which is the key for precise cell tracking in studies of migration. And some of these features are really unique and needed for the migration studies. There's the label-free, that you study the migration in native conditions, where the cells are not affected by the technique that you are using to study them. The data are quantitative, so you can study how they move and what happens with the dry mass during the movement. When you want to visualize really fastly or highly motile cells, the fast acquisition is something that you want from the microscope. And high contrast, once again, I already said, it's really needed for precise segmentation, precise cell tracking. And if you don't want to waste your time with the analysis, you can load the data into some AI-based analytical software, and the QPI data are really suitable for this. The special advantage of our microscope, the Telite Q-phase microscope, is that you can even study the migration in the scattering media. So that was all from my side today, and